Okay, does it show recording for you?
Yes.
Okay. Hi guys, I'm Isha Thapar. I'm the editor-in-chief of Scientia, and here I'm with Mary and Sai, co-authors of Discovering Phage Casserole using microbiology techniques to isolate an Arthrobacter bacteriophage. So I'm going to ask them a couple of questions about their research. So what made you guys interested in being part of the C-Phages program?
Well, we're both interested in biology, and Dr. Phage's program is just a great way to get involved with microbiology and have a hands-on experience. So it was really amazing.
Yeah, definitely. I think something that was so exciting for me is to get involved in research in freshman year, and this course allowed it with Dr. Dare. And overall, it was such an enriching experience for us.
Awesome. And then can you summarize the steps of phage discovery?
Yeah, so the first step is isolation, which just involves collecting a soil sample. And then next comes purification, where we wash the soil sample and we want to ensure through plaque assays that we only have isolated one phage.
Yeah, and after purification, where we confirm that we have one phage, is amplification, where we have a small sample of that phage and we essentially amplify it so that we have a large content to work with. And once we have a large amount of that phage, we undergo the characterization process where we discover what type of phage it is. And that involves like the cluster and the different properties of that phage. So specifically Phage Casserole, it is part of the AV cluster. And something exciting about that is that it is the fifth, right? Or sixth?
Or sixth. I think it's
I think it's the sixth ever discovered AV cluster phage. So that was really exciting about this.
Great. And then bacteriophage casserole is now available in the Actinobacteriophage database. What is this database used for?
Yeah, so the Bacteriophage database is a place where like everyone who discovers a phage can record all the data about it, and that shows you where the lysates are stored, so other people can use that information to learn more about bacteriophages. So it's just a place where everyone can access the same information and add to it if they learn more.
Awesome. And then what's the process from here? Like, what are you doing now this semester?
Yeah, so last semester where we ended was that we have samples of the phage DNA and it was sent for sequencing. So the sequencing aspect would tell us what specific genes are within that sample. And this semester it's all in silico, which is bioinformatics. We would go into a program called DNA Master and we would sift through each specific gene and ensure that it is correct. And that's an entire class project now. Due to the amount of genes, our entire class is going to work specifically on Phage Casserole and identify each gene, and at the end of the semester we will have completely sequenced Phage Casserole, and that will be again available in the Actinobacteriophage database, so for future researchers to use if Phage Casserole is useful to them.
Great. And then what was your favorite moment in the C-Phages?
I think my favorite moment, so like the whole semester, you're like working with the lysates and looking at your plaque assays, and at the very end, you're able to use electron microscopy to like see what it looks like. And so I think after working on it for so long and then finally seeing like a computer image of what Phage casserole looked like, it was just so exciting, and I think that was my favorite part.
For me, I think both of us had these moments in lab and we were like, oh my gosh, we have no idea what we're doing. And we're just like picking different things and we're like, okay, let's do this and see if it works. And I think we did some crazy things like that where we like did something where everyone would do once. We did that 44 times and it ended up working. And yeah, those are some good times.
And lastly, my favorite question, why did you name your phage casserole?
Well, so we had like another name originally, and that's what we called it like the entire semester. And then we finally submitted the name, and it got rejected. And we had like 4 days to come up with a name. We like called my friend from middle school, and we were like, "What do we name our page?"
Like anyone would do.
Obviously, and it was a week before Thanksgiving, and you know the name casserole came up, and it felt right.
In the moment, it was everything we could have think of.
Awesome. Thank you guys so much.
Thank
Thank
you,
you.
Isha.